Hey guys, my name is Max and welcome to Simple Biology. Today we're going to be talking about transcription and translation. Now, if you remember back to our cells unit, I mentioned these two processes briefly when we talked about ribosomes. Transcription and translation are the process by which the information in your DNA can be transferred into the information that will be used to create your proteins. So every single protein in your body is going to rely on both transcription and translation in order to be made. So that's why these two processes are so incredibly important. So the first part, of course, is transcription. And so transcription is the name we give for mRNA synthesis. Now, mRNA is messenger RNA, M is for messenger, and RNA is just a type of nucleic acid that is slightly chemically different from DNA. And so the process of mRNA synthesis, which we know as transcription, is the first step in this process. So the first thing we're going to look at is what's known as RNA polymerase, and this is a very important enzyme, just incredibly important to life, and it's essential to creating these mRNAs from the DNA. So what it's going to first do is it's going to bind to a sequence known as the promoter. It's this little green portion of the DNA. These two lines are DNA, and this green part is the promoter. So after it's bound to the promoter, the DNA will be pulled apart by the RNA polymerase, and it will be used as a template for the RNA polymerase. And the RNA polymerase goes through this DNA, it's going to be using the same coding to create a version of it as mRNA. mRNA will be exactly the same as the non-template strand, which is the one that's not being used to be coded, but there will be a slight difference in that it uses U, which is uracil, as one of its bases instead of T, which is thymine. That's another one of those slight chemical differences between the two nucleic acids. So I'm going to go back to one little part that I mentioned, and that mRNA is the same as the non-template strand. This may seem a little bit weird because it's using the template strand as the template, but when you realize that it codes inverse, so A will be U, and C will be G, and G will be C, T will be A, that means that it's exactly the opposite of the template strand. Now, of course, if you remember about DNA, each strand is the exact opposite of its partner. So that's why we use the non-template, because it'll be identical in sequence, except for that one little difference. Next, we have translation, of course. So translation is protein synthesis, which occurs in ribosomes. That's why I mentioned ribosomes at the beginning of this video. Essentially, what happens is that the messenger RNA will bind to a free ribosome, and it'll start going through, and these little blue things, which are known as tRNA or transfer RNA, will bind one amino acid at a time to extend a chain of amino acids, which is known as a polypeptide. As it goes through, it's interesting to note that it's not just one base pair per amino acid. There will actually be 3 base pairs that code for each amino acid, and so that's why you can have 20 amino acids with only 4 base pairs. With certain ribosomes, depending on what protein it's creating, they'll then go and bind to the rough endoplasmic reticulum, and what that does is that it initiates a set of processes that will lead to that protein being excreted or put into a vesicle or several other places. Lastly, we're going to go into some mutations that can occur with mRNAs and just anything along this process. So these are mutations in specific genes rather than mutations in whole chromosomes, which we have gone over before. Don't get those too confused. The first we're going to have are silent mutations. And now if you look at a codon chart, which is it's the genetic code of each three base pairs and which amino acids they code for, there are some doubles. There is some redundancy in that code. So a silent mutation is one that doesn't actually affect the amino acid sequence, just the base pair sequence. And so what this will do is it will change usually the third amino acid in a codon. A codon is a triplet of base pairs, and I said that wrong. It'll change the third base pair in that triplet in that codon. And by doing so, if it changes it just right, it may not actually affect what amino acid it codes for. Next we have missense, which is when one or more amino acid is altered. This is generally when that redundancy doesn't kick in, and maybe the first or second base pair is altered. And what that will do is it'll change just that one amino acid to a completely different one. Otherwise, it might change. It might delete or add an extra base pair, and this will result in extensive missense, which we refer to as frame shift, and I'll be getting into that in a minute. Next, we have nonsense, which is when a certain deletion or addition leads to the presence of a stop codon before it should be, so a premature stop codon. And the stop codon is a 3 base pair sequence that codes for the polypeptide to end. As you would imagine, I'm sure, if there's a stop codon before there's supposed to be, that will cut off half, maybe 2/3 of the polypeptide, which will greatly alter its function. Next we have frame shift, like I mentioned earlier, and this is anytime there is an inserted or deleted base pair. And what this will lead to is that the reading frame of those codons will be altered by 1. So every single codon from that point forward will be altered in a way that will force the ribosome to not be able to read the mRNA properly. And the last thing we're going to talk about is this one interesting thing, and that's if that you have a triple insertion or deletion that won't actually result in frame shift because it's a whole codon missing or added, but it will result in an extra or missing amino acid rather than a bunch of missense. And so that's really all we've got for transcription and translation. They're as simple as that.